The next section that we're going to talk about is mutations. We talked about gene expression, how genes are transcribed and translated into proteins or in the physical representation of the genetic information. But now we're going to discuss what happens when this process goes awry and causes genetic mutations. Genetic mutation is a permanent change in a sequence of bases in DNA. The effect on the gene mutation can range from no effect at all to a complete inactivity of the gene. And there are two types of mutations. There's germline mutations. Germline mutations occur in sex cells and can be passed down from one generation to the next. Somatic mutations only occur in somatic cells or body cells. These are not going to be passed down through generations and typically only affect one region of the body. They can still be fatal though. For example, they may lead to cancer. So germline mutations are found in your gametes, and they're going to be passed down from one generation to the next. If, however, you were to expose yourself to a great deal of sunshine and develop cancer because you've overexposed the skin in your arms to UV radiation, are you going to pass cancer on to your offspring? No. That's kind of like saying if you were to get into a car accident and lose your leg, are your children going to be born without a leg? No. So autosomal mutations, or mutations that happen in somatic cells, are not going to be passed down from one generation to the next. That only happens in germline cells, only when they are passed down through the gamete. So as we've mentioned before, DNA is super remarkable in replicating and coding accuracy. However, mutations can occur into the DNA once it's been made. And the problem with having any error in your DNA is any mutation that presents will get passed on to every strand of DNA that is synthesized from that point on. Since your skin cells are constantly replacing and they're constantly undergoing mitosis, if you get a mutation in your skin cells, that mutation is then going to be passed on to all of the resulting skin cells that come after it. So there are a variety of changes to DNA that can alter structure and functionalities of DNA going forward. The first type of change that we're going to look at is a chromosomal change in structure. This happens when whole sections of the chromosome are duplicated or deleted. And we've already kind of talked about a few of these. So we've already talked about a few of these, so I'm going to let you spend some time on that later. There are also gene mutations. These occur on a much smaller scale. They're only going to cause changes to one or two nucleotides. However, this can still have a drastic effect on the overall gene structure. So we're going to just take a quick look at this. So here we have an original strand of DNA. If, however, we accidentally make an incorrect pairing and pair a cytosine with an adenine instead of a thymine, we're going to see a change in the amino acid. So the potential outcome is that what originally was CAT and coding for GUA, which should have been a valine, is actually going to code for, we've had one tiny little base pair change, but when we've made that, we now suddenly have CGT instead of CAT. This is going to code for alanine instead of valine. So we have changed the amino acid. Now, a point mutation involves a change to a single DNA nucleotide. So one type of point mutation would be a base substitution. We're just changing one codon that's going to have a different permanent change to its DNA. So it's called this because, so it's a point mutation because it involves changing a single DNA nucleotide, and that change is going to alter the transcription and can potentially change the sequence of the amino acids produced by translation. This type of point mutation is sometimes called a base substitution, where one incorrect nucleotide is substituted for the correct nucleotide. This can lead to disastrous effects if the amino acid encoded by this new codon, if it changes the amino acid that is coded for by this new codon. Or it could potentially result in a stop codon that truncates the protein early. There's also the potential that nothing will change. Sickle-Cell Disease is caused by a point mutation that results in a codon for valine instead of glutamic acid. This single amino acid change alters the shape of the red blood cells. Instead of being round and smooth, they become rigid and sickle-shaped, which can lead to clogs in veins and general decreased blood flow. Here we have a single base substitution where we have substituted out a G for an A. what we're going to find is that resulted in changing the amino acid sequence from methionine, phenylalanine, threonine, arginine, isoleucine to methionine, leucine, threonine, arginine, isoleucine. One tiny point mutation, one tiny amino acid substitution. This might not be so bad. If we substitute a hydrophobic amino acid for another hydrophobic amino acid, we might not see a drastic effect. If, however, we were to substitute a hydrophobic amino acid for a hydrophilic amino acid, we suddenly could drastically change the shape, completely change the shape of the protein. And so a point mutation can have anything from no effect to disastrous effect. And those are point mutations. Oh, I seem to be missing a slide, so I'm going to pause for just a moment. All right, I seem to be missing my slide on frameshift mutations, so I'm actually going to leave that to you. Spend a little bit of time going over your chapter about frameshift mutations, and we will discuss it in our evening discussion groups. That's it for this section.